Myself, Dr. Ajay Kumar, Assistant Professor, Galgotia University. We will discuss the topic History Accounts of the DNA Discovery. Structure of the DNA.
In the mid of the 1800s, the disciplines of the genetics was born with the work of the scientist Gregor John Mendel, who studied his experiments by using the plants pea, named is the Pisum sativum. In the early 20th century, Mendel's work was rediscovered and further by the scientist working with the fruit flies and the plants such as the corn. The use of microorganisms as the models for the genetic studies was also followed. The microorganisms, especially the bacteria, was the ideal models for these experiments because of their unique characteristic features. One important feature is the nature of their genomes. The term genomes refers to all the DNA which present in the cells or the virus. The prokaryotes normally have one set of the genes, that is, they are the haploids. In addition, they often carry the extra chromosomal genetic material known as the plasmids. The eukaryotic organisms, including the eukaryotic microorganisms such as the fungi, usually have two sets of the genes and called the diploids, and they rarely have the plasmids. But the viral genomes are significantly differ from the other cellular organisms. Now the genetic materials is that substances which not only controls the inheritance of the traits from the one generations to the next, but is also able to express its effects through the formations and the functionings of the traits. It has the following properties. Its hereditary informations must be present in the coded forms. The structural elements must be ubiquitous in their distributions and they are able to replicate and they should be same both in the quality and the quantity, and the more important properties is the stability. Now DNA as a genetic materials. DNA contains the genetic informations which are the specifying the biological development of all the cellular forms of the life and many viruses. DNA acts as a genetic material and used to store the genetic informations of organic life form. The DNA encodes the sequence of the amino acid residues in proteins using the genetic code, which is the triplet code of the nucleotide. There are the two evidence that supports the DNA work as the genetic material. These experiments are the indirect evidences and second is the direct evidences. In indirect evidence, we studied the Griffith experiments, and the direct evidence, the experiment is known as the Hersey and the Chase experiments. Griffith experiments. The early work of the Fred Griffith in 1928 on the transfer of the virulence in the pathogen Streptococcus pneumoniae, commonly called the pneumococcus, set the stage for the research and showing that the DNA is a genetic material. He studied the two different types of the bacteria. One is a smooth and second one is the rough. These bacteria cause the disease, which is generally known as the lung disease and well known as pneumonia. The Griffith, who used the two different strains, one is the type 1 smooth strains and second one is the type 2 rough strains. The type 1 smooth strains have the properties of the virulency and have the lipopolysaccharides capsules layer and could kill the mice by causing the disease pneumonia and made the round colonies on a culture plates when we cultivate. Whereas the another strains, that is type 2 strains, is rough strains, which was the avirulents or the non-virulent and lacked the lipopolysaccharide capsules and grow in the rough shaped colonies on the culture plates when we cultivate. In these pictures you can see the Griffith experiments on the mice, and there are the two different types of the strains of the pneumococci. One is the rough, second is the smooth. In this picture you can see that the mice is injected with the rough strains, which is represented as blue in color. After the injections of the rough strains, the mice remain live. In a second case, when the smooth strains of the pneumococci injected into the mice, which is round in shapes, after the injections of the smooth strains, mice becomes dies. And in the third one case, when the smooth strains of the pneumococci are the heat killed and are injected into the mice remain live. And in the last and the fourth cases, when the rough strains and the heat killed smooth strains of the pneumococci are the mixed together and injected into the mice finally dies. Now the Griffith found that after the performed of his experiments that the mice are killed, but Griffith was unable to explain the reasons behind these results, but he called this change of non-virulent bacteria into the virulent or the pathogen is a transformation. Now the Griffith found that after the performed of his experiments that the mice are killed, but Griffith was unable to explain the reasons behind these results, but he called this change of non-virulent bacteria into the virulent or the pathogen is a transformation. After the experiments of the Griffith, there were three scientists, name is the AVERY, McLeod and the McCarthy, who performed experiments in 1944. He set out and discovered that the constituents in the heat-killed virulent pneumococci, which was responsible for the Griffith's transformations. These three investigators selectively destroyed the constituents in the purified extracts of the virulent pneumococci, means smooth cells, by using the enzymes that hydrolyze the DNA, deoxyribonucleic acids, RNA means ribonucleic acids or the proteins. They then expose the non-virulent means rough strains of pneumococci with the treated extracts, and then the transformations of the non-virulent bacteria was blocked only if the DNA was destroyed, thus suggesting that the DNA was carrying the genetic informations required for the transformations. The publications of these studies by the AVERY, McLeod and the McCarthy in 1944 provided the first evidence that the Griffith transforming principle was the DNA, and therefore the DNA carried the genetic informations in bacteria. So in these experiments, AVERY, McLeod, McCarthy explain that when we use the rough strains and inject it into the mice, the mice remain alive, and when we directly injected the smooth strains into the mice becomes dead. But when we heat killed treated smooth strains injected into the mice remain lives. And when the heat treated smooth strains mice are mixed with the enzymes, mixed with the rough strains, then mice becomes died. And when the heat treated smooth strains and the rough strains bacteria are the mixed with the enzyme DNAs, then the mice remain lives. And in the fourth cases, when the heat treated smooth strains and the rough strains are treated with the protease enzymes, and then these mixtures are injected into the mice remain, the mice becomes dead. The reason behind this, when we use the DNase enzymes, the DNase enzyme degraded the DNA materials in smooth strains and transformations not occurs. Whereas in the fourth case, where we use the protease enzymes only degrade the proteins, not the DNA of the smooth strains. Then in this case, the DNA transform from smooth strains to the rough strains and the mice died. So by these experiments, AVERY, McLeod and McCarthy explains the reason behind the transformations. After the experiments of the Hersey and experiments of the McLeod-McCarthy, the two scientists were the Hersey and Chase in 1952 proved that the DNA was a genetic material in the bacterial virus known as the T2 bacteriophage. That time, some luck was involved in their discovery, for the genetic material of the many viruses is the RNA, and the researchers happened to select a DNA virus for their studies. Both the scientists used the bacteriophage in his study, and he labeled with the help of the sulfur and the phosphorus elements. The controversy surrounding the nature of the genetic informations might have lasted considerably longer than it did. Hersey Chase made the virus DNA radioactive with the help of 32 phosphorus, or they labeled its proteins with the 35 sulfur. They mixed radioactive bacteriophage with the Escherichia coli and incubated the mixtures for some times. The suspensions was then agitated violently in a blender to shear off and absorb the bacteriophage particles. After the centrifugations, radioactivity in the supernatant where the virus remained versus the bacterial cells in the palate was determined. They found that most radioactive protein was released into the supernatant, whereas the 32 phosphorus DNA remained within the bacteria. Since the genetic material was injected and T2 progeny were produced, the DNA must have been carrying the genetic informations for the T2 bacteriophage. So that were the three experiments by which the scientists proved that the DNA act as the genetic material in all the organisms, and subsequently the studies on the genetics of the virus and the bacteria were largely responsible for the rapid development of the molecular genetics. So by these experiments it was proved that DNA have the genetic informations and act as a genetic material.